We have students do 3, 4 in a whole range of year levels, and it depends on the school and the choice is just what year level students are in. So just wanted to get a feel of what level my audience is, my young people out there who are taking this journey with us this year. It's a great subject, 3, 4 biology. See, this is what I expected, and this is what I find in my classrooms, that we've got 49% of year 12s and 51% are year 11. So it always is about half and half, which is fantastic. Students, some schools do accelerate, and there is a possibility for year 10s. Well, that's fantastic. So as I say, my name's Juliet Anstey, as I've got written on the sheet there, and I'm going to guide you through the continuation of our Unit 3 study. My presentation method is probably very different to Veronica's, but I think this is a good thing. This is why we do alter the teachers, so that you get the feel of different formats. Your teacher will introduce Unit 3, 4 biology over the year to you in a different format as well. All right, so we should be working from our School for Excellence book. I hope you all have that in front of you. Veronica introduced you to the fact that we've got 2 areas of study for Unit 3, and she was starting off with Area of Study 1. I call it the Important Molecules Unit. It's about nucleic acid, it's about proteins, and it's about their relationship. And she would have finished going through with you the idea of that relationship between nucleic acids and proteins. What I will then continue is talking about how that knowledge that has been gained is used in research. So yesterday, this is what Veronica did with you. She went through a lot of the key ideas about those important molecules, the nucleic acids and the proteins. You need to obviously know their structure. You need to know the relationship, protein synthesis, all that idea. And in particular, the concept of genes, the structure of a gene, what a gene is, because that's what I'm going to be doing with you, taking the gene knowledge a little bit further. So I'm going to go through these last point here about how genes are regulated in the cell, because that's important knowledge to then move into the second part of this Area of Study 1, the tools and techniques idea, the use of knowledge to use in today's society and research. So that's where I'm taking you in this first lecture. If we have time, we then might click into Area of Study 2, the second area of study, but I don't want to rush this, because when you get to school, these 2 important parts about the relationship between proteins and nucleic acids and DNA manipulation, they're going to be the key focus that your teachers will be giving to you in term 1. Now, having said that, I believe you've done a few questions, but looking at the page, there are some questions in our book, and I hope you'll have a chance to do them over the time as part of your revision. You can go back into these notes and try the questions, and the solutions are at the end. But there's just a couple I'd like you to have a go at based on yesterday's work, just to see if you've got your mind thinking about what went through yesterday. All right, so let us have a look at these sort of questions. Now, we get a lot of information, and what VCAA loves to do is put diagrammatic representation rather than lots of text to read. They often have diagrams, especially of processes, and this is a typical example of a diagram. So when you look at this, you should be able to recognize immediately what is showing in here. Things like, obviously, this is the cell membrane, this the nuclear membrane, this would be endoplasmic reticulum, things like that. So what we've got is a real highlight of some process happening within the nucleus, the events associated with the production of a polypeptide chain, which is what a protein is. So that's what this diagram's showing. Now, what we have to do when we're answering these is to make sure we are reading the correct information. So what is the name Now, when you're asked for the name of something, all you have to do is write the name down. You don't have to write the name of the process at step 1 is. I know in school we've always encouraged you to do that, but what we're talking about here is just write the name down of the process in step 1. As I read questions, I highlight, underline key aspects of it so that I'm focusing. They're talking step 1. So this is where, in this picture, we're going from the double-stranded helix, the double helix DNA, into a single-stranded molecule. To go from that, we obviously should know the name of the process is transcription. That's the answer. Now, the next one is the name, again, of the product. The first was the process name. This is the product name. So it's asking you, what is that product called? Now, if you have a look at the picture, we've got product 1 after 1 and another product after step 2. So here we had to be very specific because the DNA is transcribed into, first of all, pre-messenger RNA. Or you could write it out fully, pre-messenger RNA. It is acceptable to write an abbreviated form when it is an accepted abbreviation rather than write the full word messenger. And you don't have to write ribonucleic acid. RNA is an accepted abbreviation. But if you just write messenger RNA, that is not correct. Because, as it says, there is step 2. Now, what a lot of students do is they see part 1 is step 1, so they then read part 2 of this same question as asking what is happening in step 1. That's not what the question is. It's asking you what is happening in step 2. And step 2 is where we are going from the pre-messenger RNA to the messenger RNA. And as you can see in the picture, the pre is a long single wiggle-waggle line. At the end of 2, we've got a shorter wiggle-waggle line. So something has happened to make the pre-mRNA into a shorter molecule. And we're asked just to talk about what occurs. Now, it says outline. It's going to be 1 mark worth of information, so we don't need a great big explanation. So what you would be talking about is the pre-messenger RNA has the introns, which are, remember, the non-coding sections, removed, and the exons, which are the coding sections, joined to form mRNA, or you can write mature mRNA. So it's just outlining what's happening. Now, I'm not saying just that the introns have been removed. I'm then saying the exons are being joined because we have to actually form this mRNA molecule. So that's what's happening. It's introns removed, exons joined. Now, there are other responses that can be then put on. You could add talking about the methyl cap and the poly-A tail. But really, the main key event that is happening between the pre-mRNA and the mRNA is the removal of those introns. We know they can be put together in a whole lot of different order called alternative splicing, but that's a different process. So really just talking about step 2. And again, for 1 mark, you don't need to go into lots of detail about enzymes and all those sorts of things. Although as you move through your course, you will learn a lot more detail there. All right, moving on. Name the event occurring at structure 3. So what is happening here? What is happening here is obviously that mRNA is being read by the ribosome, which is this funny little squiggly thing here. And we have this little dotty thing made, which is the polypeptide chain. So to name the event is in fact translation. It's not asking you to explain. Explain would be to go into the detail on the process. This is a name. Right, next one. Name, again, structure 4. Here's structure 4. Make sure you're reading the correct number and looking at the correct part. Very easy in a SAC or an exam to be a little bit nervous and to misread. So structure 4. The structure that is at 4, that is being highlighted there, is transfer, not transport, RNA, or tRNA, whichever one you wish to write. Transfer. So common students write transport because it does transport, but I don't use that word. I say carries amino acids. So the function of this structure, and as we know, it's going to carry the little amino acids, the specific amino acids. So it's going to carry the specific, that's a very important word, amino acid to the ribosome to join its anticodon with the complementary mRNA codon. Now, that's a lot of information. Again, 1 mark. It probably is enough for you to have that as your answer, as long as you have the word specific. But VCAA, and I suggest if you can, what I call modify, if you can embellish your answer a little bit and add a bit more information about the anticodon on the tRNA being complementary or opposite to the codon on the mRNA, just adds to the quality of your answer. You don't get an extra mark for it, but if VCAA wants that extra information, you've got it there. So always try to just see if you can just add a little bit of extra information, relevant, not irrelevant. So I haven't gone into the amino acids joining by peptide bonds. That's not relevant. But this is about tRNA. And the function of tRNA is to carry that amino acid. But the reason it's that tRNA is because of its little anticodon. And its little anticodon, which is this bit here, is going to be complementary to the codon that the ribosome's reading. So you can see how you've got a standard answer. Oh, just that quality. And that's what we're after. And that's what your teachers will be encouraging you to just improve that quality of your answer as you do those sorts of questions. So from one diagram, a whole lot of information can come out of it. Hopefully you did okay with that. And again, if you didn't know or couldn't answer all these things, this is your first time for a lot of you at seeing this information. Therefore, as I say, when you get back to school, you'll have it again and again and again, and you'll be a lot more comfortable in these answers.